Hello everybody, this is Nagarajan working as an assistant professor in MAM School of Engineering Trichy, aggregated by NAC, approved by AICT and affiliated to Anna University, Chennai. So the previous class we were discussing about the different blood cell counting techniques and that is hematocrit method, and now we are going to study the manual method. Okay, so manual method is an tedious process. Let me explain that one in detail. So manual method, as I said, it is a tedious process. It is done using the microscope. Okay, especially in this method, electron microscope is preferred, so that it will be easy for them to capture the image and they can go for continuous validation, that is measuring. If it is a normal microscope, then it will be very difficult. Okay, it will be very difficult, and if the technician miss the count, then it is again he has to go for the next iteration. Okay, that is next sampling process. So if we choose electron microscope, then we can take the image, capture the image, and we can go for counting. Okay, so microscope is utilized in this technique. First, the blood cells is diluted in the ratio of 1:10 or 1:200 for counting RBCs. Okay, 1:10, sorry, 1:100 or 1:200 for counting RBCs, and the ratio of 1:10 to 1:20 for WBC. Okay, so the diluted blood is then brought to the counting chamber of 0.1 mm. Okay, 0.1 mm, which is divided into a number of squares, more number of squares. Okay, there will be a more number of squares, and the technician should count the number of cells present in each squares, and using the square techniques, that is using the number of squares, they will do the calculation to say the exact value. Okay, it is magnified about 500 times, and the number of cells present is determined. Okay, in this, as I said, it is a tedious method, time consuming, but still it is used in many small hospitals for measuring these techniques, for measuring the blood cells. So here you can see a square. Okay, here you can see glass plates where the cells are, where the sample is placed. Okay, so if the cells are one third of the, at least one third, you can say, sorry, not one third, at least 3/4 of the cells should come into the square, then only it will be considered. If 3/4 is outside the line, then it won't be considered. So here you can see 1, 2, 3, 4 chambers are there. Okay, so the counting will be taken in these chambers, and listen, this is the chamber. Okay, one of the chamber in this entire slide. Okay, in this entire test slide. Okay, so by this way the slides countings will be done. Okay, let's next I will show you a small video how the measurement is done. Okay, so that you can able to understand clear. Just a moment.
Use a cell counter to tally the number of viable and dead cells. On a worksheet, write down the total viable cells, total dead cells, and total cell count. When you're looking through the microscope, you'll see a chamber with 4 corner squares to count. There are 2 chambers, so a total of 8 corner squares, 4 per chamber, will be counted. This is a graphical representation of the counting grid within one chamber under 40x magnification. This image represents an actual shot of one of the corner squares at 100x magnification with hepatocytes in view. The viable cells are clear in color, the dead cells take up the trypan blue and are blue in color. To count precisely, it's important to understand the role of the limit lines, the triple lines that outline the sides of each corner square. To ensure that cells are not counted twice or overlooked, only include the cells that fall on 2 of the 4 sides of each square. For example, include cells that touch the top border and the left border, but exclude cells that touch the bottom border and the right border. When counting the cells on the limit lines, the cells must be touching the center limit line to be included in the count. If the cell is only touching the outer limit line, then the cell is not counted. In this illustration, the darkly shaded circles represent the cells that are counted. You must maintain the same limit line rules when counting other squares on the hemocytometer. Once you've focused your microscope on an outer square, start your count at the top left-hand corner and follow the directions shown by the arrow. It is important to count the 4 corners in both chambers for a total of 8 squares counted. The number of cells in each corner square may vary.
So in this it is clearly said that there are 3 lines. Okay, when the cells touching in the outer line, that is not into accounted, and when the cells touching at least the middle line and the inner line, okay, that is taken into account. That is the maximum limit of taking into account. That is why they have given 3 lines in all the 4 sides. Okay, so after this has been decided, the cell counting must be taken from the left to right, and then from right to left, and then from left to right, and then from right to left. Okay, so this you can he is very clearly explaining, and this is a wonderful session. We will continue this one.
On your worksheet, tally the total number of viable cells, total number of dead cells, and total number of cells counted. To calculate the percent viability, divide the number of viable cells by the total number of cells counted. Multiply that number by 100 to get your percent viability.
Okay, I think now you might have come to a conclusion and got an idea how the blood cell counting is done manually. Okay, usually this type of blood cell counting is done to identify the WBCs. Okay, because RBCs can be determined exactly in the hemocrit method, whereas in that you might have seen very low, very minimum quantity of WBC and platelets in that capillary tube. So this method is best suited for measuring the WBCs, and also it will help to determine the other cells also, but for WBC it is recommended. Okay, hope this class must be very useful for you, and we will see with next topic, that is laser based red blood cell, that is laser based blood cell counting techniques, in the next class. Okay, so thank you.